The clinical data from a trial to commercialization is the ultimate goal of a bridging study. And so the question is, how do you get from point A to point B to point C for IVD development? Well, point A is where you start. You've got your investigation use reagents. You've used those to generate the clinical data during the trials. The reagents were manufactured on a small scale, just enough to run the clinical trials. And you had designated testing personnel who were specifically selected to participate in the trial. So that's where you start off with. You've got your clinical data now from your small-scale IVD reagents, IUO reagents. The next point is somewhere during that trial or during the FDA review process, somewhere along the line, the assays have been modified, either to better purify, to improve the yield of the reagents, but the reagents have been modified, and you need to understand what those modifications, what the impact of those modifications were to the clinical data that have already been collected. Point C is now you're ready to launch. You've launched the product. This is your final IVD assay. You've got FDA approval or clearance to market it. You're now marketing it, manufacturing it on a larger scale, and you are making improvements to the manufacturing or to the assay. And so there's an FDA submission amendment for those modifications. So how do you get from point A to point B to point C in the pre-launch phase? And that's where these bridging studies come in. The bridging studies to go from the small-scale IUO reagents that were used to generate the clinical data through modifications during the process, if there were any, and then to the final assay that's launched. So now we're going to talk about some examples of pre-market bridging studies for IVDs. And we have three examples, one of which hits at point B, and the other two address point C. Remember, point B is the modifications. So a pre-market or pre-launch bridging study for modifications, the purpose of that is to demonstrate equivalence. So you have to do equivalence studies for that. At point C, you're now with the final product, so you need equivalence studies for lot-to-lot manufacturing. And that's especially critical if the reagents haven't been made recently, if there's been a lag time between the final lot you made for the clinical trials and your launch lot. And then for a companion diagnostic, this is very critical too, demonstrating the equivalence of the clinical trial data to the final commercial assay. And that's critical for companion diagnostics because usually the drug submission is taking longer than perhaps the FDA, the IVD submission is taking. So the trials are a little bit longer, so there may be a long time between the research use or investigational use reagents that you made for the companion diagnostic trial versus when you're finally launching it. So here's the first thing we think about when we've got a pre-market assay modification. You have to look at the performance risk, the business risk, and both. Why are you making the modification? Is it on the continuum of risk, is it an enhancement or is it due to a failure? If it's due to an enhancement, that the manufacturing people have found a more efficient way to make it, to improve yields, to reduce background noise, whatever it is, it's an enhancement. And as such, there's a longer time before you need to go to the FDA. You do design control, you handle the changes through design control, and then you implement all the changes. You do a clinical validation of all the changes, and then you submit to the FDA. And how many that all changes is before you go to the FDA is a tough call to make. It depends upon the type of change, the impact of the change, and how many changes you've done. And that's a gut feel from the regulatory group, so it's hard to put a number on that. From a failure perspective, that's an easy one. If it's a failure, you've got to implement the change, and you have to repeat the validation and submit to the FDA.